Hello to everyone, wherever you are in the world. I am Tom Royston. I'm the head of the Richard and Lawan Hill Department of Biomedical Engineering at UIC. And I'm just going to give about a 5-minute intro to the department, and then I'm going to hand it back to our Director of Graduate Studies, Salman Khatani. So we are, of course, located in Chicago, very near downtown, within a mile of the Willis Tower, formerly known as the Sears Tower. If you want to, you'll find a lot more information about our department at our website, bme.uic.edu, and I have a couple links here to our annual report and to our fall and spring newsletters. A little bit of history about the department. We are one of the oldest biomedical engineering programs in the country, founded in 1965. We have BS, MS, and PhD programs in biomedical engineering, MS and PhD programs in bioinformatics since 2003. We are a department that is in both our College of Engineering and the College of Medicine at UIC. UIC is the largest college of medicine in terms of med students in the country. Our department has had a lot of growth over the past decade or so in terms of the number of students. I list here undergrad enrollment. We've also had growth in the graduate enrollment. We are, like many biomedical engineering programs around the country, we are, in terms of gender, we're pretty much equally divided, men and women. Like our university, UIC, we're also very diverse in terms of ethnic diversity in where our students come from, all over the country, all races, all ethnicities, and all over the world. I have a few of our rankings here about UIC. Let me talk a little bit more about our department, and Dr. Khatani will go into more about our graduate programs, but this is a little snapshot of our current enrollment in our bachelor's, master's, and PhD programs, showing the number of men and women. These are just some of the companies that have recently hired some of the graduates from our programs. This is a recent stat in terms of related to our research, and which is something that involves many of our graduate students on funded research projects. In the recent Blue Ridge ranking, which is something that medical schools follow, in the biomedical engineering category, we were ranked 4th in the country in terms of the amount of NIH research funding. We are, you're going to learn about, there'll be presentations by several of our faculty tonight. We have a number of different areas in the department that you may be interested in, all of them with external research funding, which will support graduate students on projects. So we have a number of faculty in our bioinformatics, quantitative biology area, a number of faculty in cell and tissue engineering, regenerative medicine area, devices and diagnostics and therapy area, medical imaging, physiological modeling, as well as neural rehab engineering, smart prosthetics. So you're gonna learn some more about what a lot of these faculty are doing, and these images I showed on these past slides. Basically, I cut and pasted or pulled these from some of our most recent annual reports or newsletters. So if there's something here that is of interest, please check out our website to get more information. And I believe at this point, I want to hand it back to Salman. And Salman, are you there?
Yes, I'm here.
Okay, all yours.
Okay. So hi everyone. It's wonderful to have you on this virtual recruiting event. So let me just give you a little bit of brief overview of what's going to happen next. I'm going to give you a real fast, maybe 5 minutes overview of our graduate degree programs. I don't want to, this is all on our website, so I don't want to belabor the point too much. And then what we'll have is we'll have 7 faculty or so give us essentially 5-minute presentations on what their lab is about, what kind of students they have, where they have placed students in the field, etc. And then we'll have a student panel. I've invited 4 PhD students, one who has graduated, and they're going to basically answer questions around, they're going to essentially answer my questions around why they chose UIC, why Chicago, etc. And then we'll have 6 more faculty give 5-minute spiels, and we'll at 8:30 p.m. Central Standard Time in Chicago, we're going to then break out to breakout rooms with the various faculty for another half an hour. And I will create breakout rooms while all this is going on. And then you'll be able to hop between breakout rooms to get your questions answered. So this is a slide deck that is probably too long for this venue, but let me just go through some of the key things here. So my name is Salman Kitani. I'm the Director of Graduate Studies. And these are some interesting facts about me. I don't think you need to read about my history, but I just want to sort of mention a few things. I am an immigrant. I've been in the United States since 1992. English is not my first language, so I speak multiple languages. I have multiple degrees in engineering, electrical engineering, and bioengineering, and I run a lab. And so I am your point of contact, along with another person that I'll introduce shortly, with regards to admissions process and in BME, helping with pick labs, etc. So I'm at your disposal. Email me. I'll do my best to get back to you in a timely manner, or certainly make sure that you get the information you need. Our alternate Director of Graduate Studies is Dr. Young Dai. She essentially manages the bioinformatics program. So the department is biomedical engineering, but within biomedical engineering, we have a biomedical engineering PhD in MS and a bioinformatics PhD in MS. And Dr. Dai is your point of contact for bioinformatics, though you can always email me and I'll be happy to find the help you need as well. So probably the most important person in all of this is Miss Susan Lee. If you haven't gotten to know her, you will if you came to UIC. She is essentially she runs the show. She manages all the undergraduate and graduate programs with regards to the various processes, etc. And she really is my partner in crime with regards to how to go about helping the graduate students succeed in our program. And then you obviously just saw Dr. Royston as well. So I'm going to share these slides with you. Everybody who registered, I will email these slides out, so you don't have to worry about taking screen captures or memorizing it. But these slides basically go through our flyers for MS program, what are the credit requirements, how many of those credits need to go to certain courses, the courses that we offer. So these slides have all the relevant links that you can use to learn more about our master's in biomedical engineering. We have a thesis option and a coursework option. The thesis option, you end up using 8 of these 36 credits towards the thesis, and you can work in a lab if the faculty is willing to mentor you. And then we have sort of analogous programs, MS programs in bioinformatics, a coursework option and thesis options, and they have some different requirements with regards to courses you need to take in bioinformatics, but otherwise they're relatively similar across the board. And then we have PhD degrees in BME and bioinformatics, and we have a couple options for the PhD degrees for people who are coming in with a master's already. Their coursework requirement is smaller with regards to number of credits. And then those who do not have an MS degree, master's degree, they obviously have to take more credits. But we have these 2 options for people coming in with or without MS. And then there's a various milestones one has to meet for the PhD degree. We do not have a qualifying exam for BME, PhD degree in BME, but we do have a preliminary exam and then a final thesis defense, which essentially constitute, along with coursework, your milestones that you have to meet. The PhD degree in bioinformatics has a different course requirement, and similar to the BME degree, people coming in with MS or no MS have different credit requirements. And then they do have a qualifying exam at the end of first year, which is based on the courses you took in your first year. And then there's a preliminary exam and a final thesis defense. So they look very similar. They have different courses. They have one extra milestone here that you have to meet. But again, all these slides, all this information is on our website. I will send this to everybody, so nothing to worry about if you didn't catch my fast pattern of speech here. You can always click these links and it will take you to the right page. So I'm sure a lot of you perhaps are wondering about what labs to select for thesis, which faculty to contact. So I get a lot of emails from students about whether we have openings in my lab, other faculty as well. So here I've kind of provided some tips that you should follow as you think about whether UIC BME is a good fit for you, which faculty to contact. Make sure you do research on what they're doing, and make sure you look at their articles. And then we have several core labs as well as affiliate labs. So we're okay with students doing their PhD in affiliate labs as long as they're obviously affiliated with the biomedical engineering department. And then these are just some tips to how to go about approaching faculty. So again, you're going to get these slides from me, so you'll be able to read this. But it's really important that when you contact faculty to work with them, that you have a sort of plan of action and not just send out a generic email, because most often than not, those generic emails end up in a junk folder. So make sure you have a very specific strategy with regards to approaching faculty, and that's what this is really about. We have several funding options, from research assistantships to fellowships, depending on whether you're international or domestic. We have internal fellowship. We have teaching assistantships that faculty get, who then award it to their students. So there's several options for funding, and all our PhD students need to be funded by a faculty when they get admitted. So that's something that's one of our requirements. And then we have several student organizations. If you came to UIC, you could participate in. They reach out to the community. They do professional development. It's really nice to be part of some of these professional organizations. And that's it. And I apologize, I went a little bit fast. I want to stay on time, but these slides will be available, and I'm at your disposal when the time comes to ask me questions. I will also be available at the last half an hour in a breakout room to answer questions about my lab, to answer questions about the program, and Miss Susan Lee will be available as well. Okay, so that's it. So what we're going to do next is we will commence with short talks by faculty. So our first speaker is Dr. Eddington, who's going to share his slides and then talk about his research. Dr. Eddington, are you okay with me recording your presentation?
Yep, no problem.
Okay, perfect. So the floor is all yours.
Okay. I can't find, oh, there it is. Okay, there we go. Okay, so my name is David Eddington. I'm a professor in the Department of Biomedical Engineering. And so my lab, we make microfluidic things. And so specifically, I have my mission statement here up top, but essentially we make novel solutions to current unmet experimental and clinical needs through applying simple devices. And so we usually leverage some sort of microscale phenomena, usually rapid diffusion or process integration. And we do this over many different scales and try to make simple devices that solve some obstacle experimentally. And so currently right now in the lab, we have two kind of main thrusts. We have a group that's working on microenvironmental control. And these are kind of the devices shown here, where we're making microfluidic devices that control dissolved gases around cells or tissues, such as oxygen concentrations or other gases of interest. And then we also control perfusion around organoids and other cell clusters using 3D printing techniques, where we're making these little bioreactor things that fit into multi-well plates. And then we also make some other 3D printed gas control devices shown here as well. And so we apply traditional microfabrication techniques as well as some 3D printing, and we're even getting into some CNC milling as well. And then the second kind of thrust of the lab is where we're applying microfluidics to microbiology, so studying bacteria with microfluidic devices. And these essentially, we're using droplet microfluidics shown here. This is a video showing two aqueous streams coming into two oil streams and creating these 10 micron diameter droplets. that we then put bacteria inside of and study how the process of genetic transformation takes place. And then this plot here is showing some complete genome sequences from this, and then here's some fancy imaging where we labeled the pili, which is this protein filament that sticks out of bacteria that's used to grab DNA and is one of the mechanisms of how bacteria take up new DNA and become antibiotic resistant, which is the public health angle of it. So in a nutshell, we work on microbiology and we work on microenvironmental control. And so how do we do it? We use the techniques that I described before. So PDMS microfabrication, where we're designing new device designs and using standard soft lithography to fabricate these in the lab. So we have a whole fabrication setup directly in the lab, spin coaters, plasma sources, UV excitation sources, and developing stations. And then in addition to this, we're also working with 3D printers, and we have a few 3D printers in the lab as well. And sometimes we use foundries to make the devices if they get a little too complicated. And then in addition to just making devices, we also make devices for the devices. And so this is some hardware development here. This is a pressure control box here. And then this spaghetti mess of wires coming out is kind of splayed out here where we're trying to harden up the system and solder in more connections to make them more robust. But we do some hardware and we do some software that integrates with the hardware with our devices as well, shown here, you know, just some standard interfaces to control stuff. And then we also do some nice imaging techniques. And here's again, this protein filament that our student Trin Lam, who I think is in one of the student groups here tonight, and you can ask her questions about, but really you can just ask her about being a student, I suppose. And so we apply all these techniques to learn new stuff about biology or you know, biological systems that in ways that you couldn't get at with any other technique. So we make new stuff to study a problem that you couldn't study the problem in the same way. And who are we? So my name is David Eddington. I've been at UIC since 2006. And currently in the group, there's about 4 graduate students. Julia, who was a master's student and will be returning again in April 2022. And then we've got Adam Schmelter, Trin Lam, and Ania Borowska. And so that's 2 kind of advanced graduate students and 1 newer grad student. And then we have a few undergrads. And then placements of current students. Recently, last one went to George Mason University, and then another student went to Evozyne, some sort of company where he's doing microfluidic things. And then previously, students have gone to USC, UC Berkeley, Minnesota, Baxter, Argonne National Labs, which is a national lab near Chicago, and Abbott, and a few other companies as well. And here's some just group shots of students. And this is the room I'm in. So this is where I am right now, actually. Okay, that's all I had. It was nice, short and sweet. And then I'm around to ask further questions about, you know, more specifics on devices or what we do. And hopefully I'll be looking for a student if everything works out. And it's nice to meet everyone. Okay.
Great. Thank you, Dr. Eddington. So again, Dr. Eddington will be available in the breakout room, so you can ask him all kinds of questions then. But let's go to our next speaker, Dr. Papatsky.
Let me share my screen here. I think this one, right? That should be presentation mode, right?
Yes, you're good.
Okay. All right. Thanks, everyone. So my name is Ian Papatsky. So this is a screen grab of my webpage. So I encourage you to, if you haven't, check out the webpage. And Salman did gave a really nice guidance on how to approach a lab or potential, you know, how to approach faculty, and a webpage is a good place to start. So my lab is another lab that does microfluidics. Dave Eddington did an excellent job giving an overview of the microfluidics technologies used in his lab. In my lab, we also use microfluidic technology, and we integrate this with sensors. and let me use the laser pointer here. Okay, so we apply the various microfluidics and microfluidic related technologies and sensor technologies to two key areas. We're interested in oncology and we're interested in hematology. And so on the oncology side, we look at liquid biopsy and using microfluidics to separate cells and fractionate samples. And we're also looking at things like cell cycle synchronization and drug response as a precision oncology application where we want to see how well the treatment or therapy options would help the patient. And so then in the hematology side, we're interested in working with whole blood and working with blood samples to separate different cells within the circulation and of the cell subtypes. And so I'm going to highlight in the next, I guess, 3 minutes or so, I'm going to give you a quick example of inertial microfluidics technology that we've been developing in our lab, and then how that could be used for liquid biopsy and blood fractionation. And so I'll show this really quick image here to tell you about inertial microfluidics. And this is I think a really fascinating area of microfluidics that doesn't necessarily deal with how the microchannels are built and structures are built, but it leverages the hydrodynamic forces of the fluid, inertia of the fluid, to impact how the cells or particles, we can also use particles, manipulated or can be manipulated within the cross-section of the channel. And so here's an example where we're putting some beads in the channel, and you can see that within a single frame of the microscope, we're taking this video with a high-speed camera. These beads are moving they enter from the side of the channel, then move close to the center. And if I stack the image, this becomes really apparent. And of course, we manipulate this a little bit. We have a really high blockage ratio, which means the ratio of the bead or the cell is pretty high with respect to the channel size. But hopefully this makes it clear to you that it is possible for cells to move across the channel cross-section as they flow downstream in the channel. So what kind of cool things we can do with this? Well, one is that we can do some really interesting studies to try to figure out why this happens and how this happens and how we can manipulate this. And so one of my postdocs, Jin Zhao, he's been doing some work in this area where we build these channels. We can look at the channels and what's going on the side of the channel from different sides. We can image the cross-section at different levels, and we can obtain images that look kind of like this, that ultimately give us a cross-section that will show that the cells or particles are moving, what they're doing in the cross-section and how they're shifting from one side to the next, depending on what the flow parameters are. And so we can look at this kind of experimental data and make really careful measurements of where these positions or where these cells are positioned in the cross-section. And what that can help us with is understanding how the forces in the flow impact cells. And we can also try to understand the underlying physics. And through collaborations with faculty and other faculty in our department, Zhengli Pan, we can attempt to model this numerically. And with numerical investigation, we can actually model or we can vary the broader range of parameters and try to replicate these experimental conditions, the empirical observations, and try to match them with numerical solutions. This understanding can really help us figure out how we can position cells within the flow. And if we can position them within the flow cross-section, we can then try to manipulate them and potentially isolate different cell populations. And so let me give you two examples. One example of where this technology could be applied isn't blood fractionation. One of my recent graduates, Prithvi Mukherjee, who just defended his PhD about a month ago, he developed a microfluidic system that you see here on the right that has multiple channels that can take a small sample of blood and separate that into blood cell subpopulations. So we were particularly looking at fractionating the sample into lymphocytes, granulocytes, and monocytes. And so this can be done in a single pass. We have this beautiful flow cytometry data. This is the inlet, and then at the outlet, you can see that the cells are fractionating into different subpopulations. And the goal here of these devices is to build a system for rapid and continuous blood fractionation without using centrifugation. Centrifugation can can present a lot of problems for in developing countries. It can present the problems with cell activation, and so this system, this type of systems, can potentially solve these problems. Another quick example of the application is isolation of circulating tumor cells from blood. And here's we're collaborating with a clinician, with Alicia Hulberg, where we're getting patient samples that we're running through our microfluidic devices, and we're able to isolate circulating tumor cells. These are tumor cells that are shed into the blood from the primary tumor in non-small cell lung cancer. And so we're able to isolate the cells. We show that these cells are larger in size from white blood cells or red blood cells. And we're able to quantify the number of cells per patient. And as you can see, we're working with just a few cells. We're literally working with 1 or 2 cells per milliliter of whole blood. The number of the cells that we're targeting and isolating is very, very small. We also able to do a post-isolation downstream analysis, and this was done with methylation on beads, looking at a particular set of genes, and we have a really nice correlation with FFP. These are preserved paraffin slides histology slides that are used to diagnose a particular stage of cancer. All right, I'll stop with this example here. Just show very quickly the picture. This is a pre-COVID picture of my group and actually Eddington's group attending a baseball game. Hopefully we can do this in the near future again. I have a couple of postdocs and about 7 students in my group right now, with a couple undergrads. My recent graduates, most recent, Prithvi, he just defended about a month ago, and he's now a postdoc at Mayo Clinic, and I've had students go into various universities and various places to do postdocs. I think I'm out of time, so I'll stop here.
Great, thank you, Dr. Papatsky. So again, Dr. Papatsky will be in a breakout room in the last part of this recruiting event. And one advice I have for prospective students is that even if you're not quite sure if you're interested in an area or not, please do pay attention to these presentations, because you never know what might strike your interest. And I think this is why the faculty are taking the time today. So it happens to be that next up is me. I'm going to talk about my research briefly. And so my lab is called the Microfabricated Tissue Models Lab. I've been at UIC this is my 7th year. Before that, I was a faculty member at Colorado State University. So all in all, I've been a faculty member for about 10, 11 years. And prior to that, I was running a company. I'd co-founded a company, and I was the director of research at a company. So I have a experience in both industry and academia. My lab is a tissue engineering lab. So we essentially engineer tissues in vitro, so outside of the body. We are able to currently do liver. Liver has been we have an international reputation in liver tissue engineering. For the last few years, we've been doing cardiac. And for the last year or so, we have been doing intestine, brain, placenta. and now cancer in collaboration with a few people at UIC. So now we're beginning to do quite a few different tissues, and we focus on primary cells as well as stem cells that we can get from patients. We focus on using biomaterials, microfluidics, micropatterning, electrospinning, high throughput microarrays to try to get these tissues to maintain their functions outside of the body. And then what we do is, this is sort of the engineering part, and then we have a whole part of our lab that applies these tissues to various applications, from modeling various diseases that I'll give you some examples of, to creating high throughput systems for drug metabolism and toxicity testing. And then more recently, we have started to 3D bioprint tissues for implantable constructs for cell-based therapies for people who are suffering from end-stage liver failure. So this slide essentially captures the entire repertoire of what we do, and what I'll do in the next few slides is give you some tangible examples so you can see some examples of projects that are going on. So one thing that we do is we use microfluidics, and you've already heard from the previous two faculty about the use of microfluidics, but these are small PDMS chips that we can use to generate these three-dimensional droplets. And then what we can do is we can take these three-dimensional droplets with cells in them, liver cells, intestinal cells, heart cells, you name it. We can then take these droplets, put them in little microwells, coat them with the second cell type, treat them with drugs. So these become sort of these 3D little microtissues or organoids. And here's sort of a video. This is, we're looking at the device from a microscope, and you can see that these microtissues are coming out. We have slowed the video down at 45,000 microtissues per hour. So it's a very reproducible technique and the use of microfluidics for this purpose. The other thing we do is we electrospin nanofibers. So we can take various polymers, natural and synthetic, and we can electrospin them in various, geometries and fibers of different dimensions to try to mimic what the cells might see in vivo. And here's a sort of a scanning electron microscope, electron micrograph of cells adhere to these fibers that you can't easily see, but they're fibers underneath. And what we find is that by arranging the cells on these fibers, we can get their functionality to be much higher than otherwise possible if you don't create these electrospun fibers. So we have projects on that. In terms of diseases, we model a very globally relevant disease, fatty liver disease and diabetes. So we study that with our model system. So here's an example where we've taken liver cells, human liver cells from patients who have passed away. And when we feed them high sugar, just like you have in diabetes, they indeed become fatty and they become insulin resistant, which is a hallmark of diabetes. Here's an example of hepatitis B virus infection. So we're obviously all living in a pandemic where viruses are king these days. Hepatitis B virus prior to SARS-CoV-2, which causes COVID-19, was the king virus and still is. It infects 257 million people. There's no cure. There's a vaccine, but there's no cure. So once people get it, they have to live with it. And we can take our liver model systems and we can chronically infect them with the virus. Here, basically, they're showing that the liver model is shedding viral antigens in the supernatant. So we can basically detect infection and use this kind of model to understand how the disease is progressing and what kind of drugs we might be able to test on it. Here's an example of our heart work. So we work in an area called atrial fibrillation, which is if you have an iWatch or any sort of smart device, it's typically monitoring for atrial fibrillation because it's the most common sustained arrhythmia globally. It's non-fatal initially, but over time, there's an increased risk of morbidity and mortality, stroke, heart failure, you name it. And again, the current therapies are not very good. So we can use our micropatterning technique and co-culture technique to organize these patient-derived cardiomyocyte cells, so stem cell-derived cardiomyocytes, which are the main cell type of the heart. We can organize them on these surfaces. We can put fibroblasts around them, and they look very beautiful. If you're not used to seeing cardiomyocytes, this is a very beautiful looking cardiomyocyte. And what's really cool is that they beat in vitro. So here's a little heart tissue that we have grown in our dish, and we can now test drugs on it or understand how atrial fibrillation is happening. The more recent work we're doing is we're creating implantable tissues for cell-based therapies. So here's an example of our published paper where we've taken silk scaffolds. So, you might sport silk on your clothing, but it turns out that silk, once the toxins are removed, is also very biocompatible material. So we can take these silk porous scaffolds, put liver cells in them, and then eventually our goal is to implant them in animals. And more recently, we got funding to do a bioprinting of a liver with multiple compartments. So my lab turns out is not this big. We have 15 people. This is actually my wife's lab and my lab combined in this picture. And our youngest member, as you can see, is quite young. No, that's just a joke. Clearly, that person is not in our lab. But my lab is very diverse. As you can see, we have people from all over the world. I think we have 11 or 12 different languages that can be spoken at any given time. Though obviously, we all speak English, so we can understand each other. And we have so far graduated 7 students, and we have a bunch graduating next year. We have placed students as a postdoc at prestigious universities, biopharma, big pharma, medical device industry, and then startups, as well as other PhD programs. So we're quite fortunate that our alumni go on to do great things, and we're looking forward to their successes. But that's pretty much it from my end. Sorry, I went a couple minutes over, but I think that seems to be the theme of today. So what I'll do next is I will go ahead and pass it on to Dr. Lee.
Can you all see my screen?
Yes, you're good.
Okay, great. I'm James Lee. My laboratory named Cell Mechanics and Signaling Laboratory. The mission of our research group is to apply biophysical engineering principles and techniques to investigate the mechanical and chemical processes in neurodegenerative diseases, with a special emphasis on the Alzheimer's disease. While we focus on Alzheimer's disease, the increased number of AD victims puts a heavy economic and emotional burden on society, and AD has become an urgent national health and research priority. So our work focuses on investigating the mechanisms and the role of mechanical alterations in AD brain. Our paper do get some attention as reflected by the number of citations. Here are a few examples. We focus on microglia, astrocytes, neurons, and the blood-brain barrier. Specifically, we study how oxidative stress, amyloid beta peptide, and tau affect cell functions. Amyloid beta peptide and tau forms unusual aggregates, which are toxic in the brain. And let me show one of the example, like cytosolic phospholipase A2 is a protein, is an enzyme I've been studying for past 15 years, and I found that it facilitates the oligomeric A beta uptake by the microglia. So the microglia, one of the functions of microglia is to remove toxic material. So we find that this enzyme actually facilitates the removal of oligomeric A beta mediated by the microglia. Here the regulation of, I hear some noise. Can you, yeah, mute it. Okay, so we can use a different kind of method. We use atomic form microscopy to study the membrane cytoskeleton connectivity. And then we found that this molecule can regulate the membrane cytoskeleton connectivity and lead to to facilitate the oligomeric A beta uptake by the microglia. Okay, so let me give you a lab tour. Our lab is located in the College of Medicine Research Building. Going through the front door of our lab, you will see a microscope room on the left and a cell culture room on the right. And going further, you will see a wet lab. In the microscope room, we have a Nikon inverted epid fluorescent microscope. This setup looks very simple, but it allows us to do some interesting quantitative observation, and usually it's quite direct. For example, we can remove cholesterol from the cell and directly observe membrane domain to become more molecularly disordered. We can also increase oxidative stress by putting some hydrogen peroxide, and the membrane domain will become more molecularly ordered. Also, we can use molecular rotor to probe the membrane fluidity at the subcellular level. We also have an atomic force microscope. This instrument allows us to perform mechanical and structural characterizations at the piconewton and nanometer level for single molecules, cells, cell membranes, tissues, and extracellular matrices. Here are the AFM image of amyloid beta oligomers and fibrils. Here is our cell culture room. All right, this is our wet lab, which is well equipped for performing animal dissection, cell isolation and purification, measurements for protein and gene expression, immunohistology, and production of elastic substrate. All my former PhD students have found their dream jobs after graduation. I currently supervise 3 PhD students and actively recruit new students. Please contact me if interested. That's it for.
Wonderful.
Yeah.
Thank you, Dr. Lee.
Okay, so shall I start?
Yes, please.
Okay, great. So my name is Shin. I'm primarily at the Department of Pharmacology and Regenerative Medicine, but I'm also core faculty at the Department of Biomedical Engineering, and I am teaching biomaterials. So my laboratory, the name is Sculpting Regeneration. It really involves combination of biology and physics, engineering and design. Before getting into science, I'd like to introduce our team. So my team consists of a diverse group of people coming from different countries. And in terms of where they're placed, it's important to mention that my first postdoctoral fellow recently became promoted to research assistant professor with his own funding from NIH, and he's looking into seeking faculty positions at the moment. And in terms of recent biomedical engineering graduates from UIC, I'd like to mention that Dr. Steven Lanzini, he ended up in Rooster Bio, which is a manufacturing company for cell therapeutic products. And Daniel Devine ended up in Culture Decadence, which is a startup company that prints the crustacean meat. And we are funded by federal funding sources. So my lab is interested in fibrosis, and it is important to mention that potential complications of COVID-19 survivors later on could potentially be the lung fibrosis, as you can see here. But fibrosis, in fact, happens across different organs, including skin, even basic scarring after injury, as well as liver fibrosis. And it is important to mention that severe fibrosis accounts for up to 45% of all death in the developed world. So this is a serious problem. Now we're trying to think about ways to correct this kind of defect in terms of the fibrotic scars, fibrotic tissues in different types of organs, mostly more focused on the lung organs. And we think that in order to treat this fibrotic scar, it is important to be able to develop microenvironments in order to precisely instruct regenerative cells in order to facilitate the tissue regeneration process in the host. And we think that we can combine biomaterial design, which offers the precise control of different types of properties, including biochemical and biomechanical. And we can combine that with microtechnology that includes droplet microfluidic approach, as well as the 3D printing approach, in order to direct the behaviors or functions of single cell or cells at the single cell level in a precise manner. as well as delivering the cells along with the designed microenvironments in order to direct the host. And highlighted publications are actually from UIC biomedical engineering students who play a very important role in the growth of the laboratory. So what type of materials do we use in order to develop microenvironments or make synthetic microenvironments in order to direct cellular behavior for tissue regeneration? You probably saw some of these materials in the context of food industry or restaurants where this material is used to encapsulate different types of liquid with different taste. In fact, this material is actually largely based on alginate, which is originally from seaweed. And what's interesting thing about alginate is that it undergoes gelation in the presence of calcium. And what's interesting about alginate here is that the reason why we are using alginate is that, first of all, intuitively speaking, it is important to note that our body is, in some aspects, like gels. That's based on some of the biophysical studies that were done previously. But it's also important to note that hydrogels can, especially the alginate-based hydrogels, can be highly tunable. It's actually used in the context of clinical cases as our own dressing. We can also synthesize or modify hydrogels in order to make them tougher. So these are very versatile type of materials that you can use not only for food industry, as I mentioned, but also for biomedical purposes. And especially in designing the synthetic microenvironments to promote the regenerative effects by these cells, it is important to be able to miniaturize these materials at the single cell level, and we use droplet microfluidics in order to achieve this goal. But as you can see, when oil is mixed with this aqueous phase, which contains cells and along with the alginate-based materials, we can essentially create the cells, individual cells that are coated with this red alginate-based hydrogels with precisely defined mechanical properties, as well as the amount of gels deposited around single cells. I wanted to demonstrate the advantage, at least one advantage of using this approach is that by encapsulating cells in very thin type of gel environment, gel coating, it is possible to be able to develop therapeutic cells along with the designer environment, if you will, into deeper tissues with narrow spaces, such as the airways, lungs. But it also provides opportunities for donor cells to restore the host environment by secreting or promoting the secretion of paracrine factors before allowing these host cells to donor cells to integrate into the host. So those are two key advantages of leveraging the so-called single cell encapsulation in order to specify the environment around individual cells, followed by in vivo delivery. This concept was recently published in Nature Biomedical Engineering, where we explored this concept in the context of mesenchymal stromal cell, or we call it MSC-based therapeutics, where in the animal models of fibrotic lung injury induced by bleomycin treatment, we demonstrated recently that by encapsulating MSCs in a soft gel coating, along with the inflammatory factor called tumor necrosis alpha, we're able to restore or reverse the established or accelerate the resolution of the established fibrosis, as shown in this histology picture as well. And the mode of action, it turns out that by specifying these MSCs with this kind of environment, we can actually promote their secretion of matrix degrading enzymes that are essential to remodel the fibrotic scars in the host from donor MSCs. So this represents an important type, at least one way to be able to modulate the fibrotic host environment by using cell therapeutics designed with specific chemomechanical type of cues. Talking about paracrine factors, in addition to their ability to secrete soluble factors as well as undergo differentiation, MSCs are also known to, and other cells as well, they're known to secrete these so-called nanovesicles or nanoparticles. They're called extracellular vesicles. And we're really just beginning to understand what they are, really. They're kind of like synthetic liposomes, which are used in the COVID vaccine, by the way, to deliver mRNA. But they're a little bit more complex in a sense that they consist of a bunch of proteins, the carbohydrate components on the cell membrane, as well as they contain a bunch of factors, some of which could potentially be relevant therapeutically. But again, as we are just beginning to understand what they're doing in the microenvironments and how we can design microenvironments in order to design or produce extracellular vesicles for therapeutic purposes. And we rather made a surprising finding earlier last year, which was published in Nature Nanotechnology, covered use and views, demonstrating that using, again, biomaterial strategy, we show that the extracellular vesicles in the tissue-like synthetic matrices, as opposed to rubber-like matrices, are able to traverse or transport through the environment, even though the mesh size or porosity of the environment is much smaller than these vesicles. And this is a unique property of the vesicles that cannot be observed when you put them, let's say, synthetic nanoparticles or just the synthetic vesicles inside this environment. So extracellular vesicles appear to have very unique role there. And you can see the transporting vesicles in the tissue-like hydrogel environment. And part of this is because of their ability to transport and permeate water through the membrane. So again, this kind of understanding would be necessary in order to really truly leverage extracellular vesicles for delivery vehicles for therapeutic purposes. With that, I'll be happy to take any question after this talk, either by email or breakout room. And that's it. Thank you.
Perfect. And I just want to remind everyone that all the faculty's email addresses are at bme.uic.edu. So please do get the contact information from there as well. So we have one more speaker, Dr. Ma, and then after that, we'll have a student panel and we'll take a short break as well. Dr. Ma, the floor is yours.
Okay, hello everyone. My name is Alma. So my lab is focused on computational molecular biophysics and systems cell biology. So the overall theme of my research is try to uncover the physical principles that govern the behaviors of complex biological systems. More specifically, we try to address two questions. One is, how do proteins work? So biology is really enabled by proteins because they can perform many functions that just not possible for small molecules. And we want to understand how proteins achieve these functions. Another question is, how do cellular behaviors show up, right? So because behaviors of cellular structures, they are fundamentally different from the behaviors of their functional building blocks, which are the proteins. And we want to know how the complex interactions between different proteins collectively gives this kind of much more significant and also regular functional behaviors of cellular structures. And all these is basically governed by physical laws. So the basic theme is to understand how do we have these complex functional behaviors based on rigorous physical principles. So on the first question, the major issue is really to understand functional dynamics of proteins, because protein functions are really achieved through dynamics. For example, enzymes can make chemical reactions that are impossible in solutions to happen very quickly, right? So how do they do it? And also such as things like allostery, right? How is a signal at remote site transmitted to the active site? And also why some mutations can significantly modify protein functions, whereas the others do not. And also, if you want to design efficient artificial enzymes, what are the guiding principles? And then what is the physical principle that determine protein structures? So the key of these is basically to understand functional dynamics, and functional dynamics are so-called activated processes. That is, the system has to cross a significant energetic barrier when it performs the function. For example, here is an example of the HIV protease, right? In order for it to work, it has to open this binding site and have the ligand come in. And this process requires the system to cross the energy barrier, which is much higher than thermal energy. So how do we understand this? The key is energy. So what we developed is so-called energy flow theory, where we can rigorously identify the potential energy through different coordinates, and also the potential energy flow between different coordinates, and also the kinetic energy flows through different coordinates. So with this kind of construct, then, for example, here, we just map out the energy flows, the potential energy flows through different backbone dihedrals of the HIV protease during its flap opening process, which is necessary for ligand binding and further function. So here we basically group these different coordinates into 8 groups, each one colored by this representative color, so that you can see, and we show it in this structure of the HIV protease. So the color that is more warm or means the coordinates has a high potential energy flow through them, and the color that is more cold means there's low potential energy flow through them. So when you see this molecule, you can see that during this base flipping process, energy flows through these beta sheets that forms the flap, and also these loops that are basically act like the joints of the flaps. On the other hand, the core of this protein dimer is relatively stable, and they experience much less energy flow during this process. And these little spheres in this structure, they are the alpha carbons of the residues that are known to have drug-resistant mutations. And what you can see is that most of these residues are located on these parts that has experienced very high potential energy flow during the flap opening process, which means if you have a mutation on these residues, it will change the interaction of these residues with other residues and changes the overall energy flow structure during the process, and will possibly interrupt the energy flow and also consequently the flap opening process. That's why they could easily induce drug-resistant effects. So the general picture obtained from this kind of energy flow analysis is that basically energy is flow from fast to slow coordinates during functional processes, and energy flows follows a hierarchical structure. And these energy flow channels, they spread through the entire molecule, making the functional dynamics collective and global. And our assumption is that the purpose of protein structure is to build these proper energy flow channels so that they can deliver, the protein can deliver sufficient energy to the reaction center. So that's the molecular part. For the cellular part, we focused on the cellular microtubule network. So microtubules are the backbones of the cytoskeleton and essential for many cellular processes, such as mitosis, morphogenesis, polarization, differentiation, transportation, etc. And because they are so important for so many cellular processes, if there's any malfunction in microtubules, then you will have disease. For example, if you have microtubule malfunction during mitosis, then you will have a neuploidy, which can lead to cell death or cancer cells. If you have microtubule malfunctions in neurons, then you will have developmental disorders and neurodegenerative diseases, things like that. So the key to microtubule function is that all the functions of microtubules are driven by so-called dynamic stability, which is the code name for the assembly dynamics of microtubules, which is a very interesting behavior that is the spontaneous switching back and forth between periods of continuous growth and shortening of microtubules. So the way our goal, well, we have a model that basically explains dynamic stability based on the basic conformational change of the tubulin dimers that is coupled to GTP hydrolysis. And with this model, we can basically explain all the known behavior of dynamic stability. And that gives us a platform to answer more functional questions like what is the molecular mechanism that the individual regulatory proteins control the behavior of dynamic stability via specific protein-protein interactions during different cellular processes. And we also can enable us to understand how different microtubule-associated proteins to cooperate with each other to achieve the desired behavior for microtubules. So that's about the research. And currently, my group has 3 graduate students, and there's another one incoming. And our previous alumni, they usually find jobs in either industries or academia. And I think I have talked enough about my research and the group. So if you have any questions, I'll be happy to answer either in the breakout room or by emails. Thank you very much.
Perfect. Thank you, Dr. Ma. So the great news is that you're getting to hear about all this exciting research. The other news, not bad news, is that we're behind schedule. So we have a student panel that I have assembled who, if maybe you can show your faces, Dr. Mukherjee, actually, who just graduated, Miss Brown, Miss Trin, and then Mr. Zhang. So they're here, and what I've done is prepared some questions for them so they can sort of talk about their experiences at UIC. Let me just go to those questions. So maybe what we'll do is I'll just call out a student name, and then you're welcome to answer that question. So one question I have is, why did you pick BME at UIC for your graduate studies, perhaps over other schools? So maybe what we can do is have Mr. Zhang answer that question, because he came to us from another institution.
Can you hear me now?
Yes.
Yeah, hi. Just very brief introduction. My name is Zhu Hao, or by Dr. Katani's name, Mr. Zhang. I'm currently a 4th-year PhD student in Dr. Ian Papatsky's lab. And basically my research right now is developing electrochemical sensors, miniature or micro sensors that can be used for in situ measurement and also point of care testing. And back to the topic that Dr. Katani just raised, why do I choose UIC and specifically the biomedical engineering program here? So just as he has mentioned, I actually previously got a master's degree in Case Western Reserve University in Cleveland, Ohio. And then after that, I decided to, work further, expanding my research focus on electrochemical sensors. And I guess the primary reason that I, choose UIC and Department of Biomedical Engineering was, Professor Dr. Ian Papatsky, because apparently for PhD studies, the research advisor is definitely very important. Of course, there are also other rankings that kind of, and also Chicago, but I think the primary reason is really about research focus. So because Dr. Papatsky, he already mentioned he does research on microfluidics as well as electrochemical sensors. So because that kind of aligned with my previous background in research back in Case Western. So I just reached out to him, and he gave me an interview and it went well. So I just hop on the bus.
Perfect. Thank you, JL. So the other question I have, maybe we can have Dr. Mukherjee answer this question. Dr. Mukherjee, how do you, or I guess you moved to Mayo Clinic, but how did you like living in Chicago in terms of the diversity, cost of living, and any other factors that you might want to talk about?
Oh, it was an amazing experience for me. Chicago, as everybody knows, is a very multicultural, multi-ethnic city. And I got the opportunity to work with different kinds of people from all over the world, and work with them in collaborative projects, be it engineering or medical. That's the scientific side. And the life in Chicago is amazing. Of course, it's such a huge city with amazing food everywhere, and you can experience the whole world in one city in a way. So cost of living is definitely much lower than most of the other big cities in US. So as a PhD student, it was definitely, what should I say, challenge-free in a way.
Awesome. Thank you, Dr. Mukherjee. So my next question is for Ms. Lam. Ms. Lam, as an international student, what opportunities and potentially challenges did you face that you think maybe people on the line may want to know? You might be muted, Ms. Lam.
Okay, sorry, I was muted. So actually, as an international student, I really care about the cultural sensitivities of the universities and also about the department. However, I think the UIC and also the BME department have done a very good job in the culture respective. So I think all the international students here don't have to worry about that. It's definitely not a challenge for you to worry about that culture. And also, like in the lab or in the university, what I worry at first is because I moved quite a lot from Seattle for my high school to Texas for my bachelor degree, and then to Illinois. I actually I was really afraid of changing the environment, and also like how well I could get along with the university, how about the people in the big city, how different they are compared to the small town that I stayed during my bachelor in the Texas. But actually in the department, I never really felt alone. Also in the lab, I got a lot of supportive. The challenge, I would say, is about like the first time that become the teaching assistant, which is like how we could explain the things to the students, and also like how to like get people really how to support them during the office hour. And for the Chicago overall, I think the biggest challenge could be the weather. During the winter, the first year was pretty tough for me, but after that, it's fine. Other than that, I think the opportunities I have a lot at the department with the faculty, and the challenge is actually we don't have to worry that a lot, except the explaining and the teaching in the department for the undergraduate student. Yeah.
Thank you, Ms. Lam. So last but not least, my question for Ms. Brown is, what advice would you give the prospective graduate students on this line with regards to how should they go about picking graduate programs, lab that they're in, that they might end up in? So, just talk about your experience being a senior PhD student.
Sounds good. So I am a fifth-year PhD student in Dr. Katani's lab. And I think the most important thing when you're trying to decide a graduate studies program, especially if it's research focused, is making sure that you're in the right lab. So making sure that you're reading some papers from the labs that you're interested in, definitely talking to the advisor, making sure that their advising style is what works with your needs, as well as it's very important to possibly talk with some of the current students in the lab and make sure that their experience is something that you're open to, and really making sure that the environment is what you're looking for in a PhD program. I've been really lucky with my lab. I've really enjoyed my experience, but that I think is the most important thing when choosing a lab to do your PhD or master's research in.
Perfect. Thank you, Ms. Brown. And, you know, as sort of all the students echoed, we are a wonderful city, the third largest city in the United States of America. It's my favorite city. I've lived all over the world. I've lived multiple cities in the US, and I'm super happy in Chicago. And, you know, it's a very accepting city. And we clearly do outstanding research, but, you know, I don't know if everybody caught this statistic that Dr. Royston put up. But this slide right here really embodies our excellence. So this is, if you look at what you're seeing here is a research expenditure by universities. So clearly Stanford is, you know, wonderful, right? 34 million. University of Illinois Chicago is right in the company of Emory, Virginia. And this is something that I just find amazing. We are doing amazing research with amazing students from all over the world. And the fact that we're able to be in the company of Emory and Virginia and Chapel Hill is outstanding. So if you came here, you can't go wrong. You'll be in a great city with great people, with great advisors, and wonderful amount of resources to do research. And this is something that I love to brag about, and I figured I'd brag one more time before you know we move on. Our next speaker is Dr. Penelope Barnaby.
Can you see my screen?
Yeah, you're good.
Okay. So my name is Beatriz Peñalver Bernabe. I am an assistant professor. I am a Dr. Ao Mao, part of the bioinformatics, part of the division of the department. And we are in the other side of campus. We are with the doctors, not that these guys are not. So I am very interested in trying to help the improvement of women's health in general. And for that, we are divided. Our group is divided in two main areas. One of us is our clinical team, in which we recruit women for our study. And then is all the computational team, where we get all that data that women have been very nice to donate it for us. And we analyze it and trying to move it into translational approaches. So we collaborate with people here in the department, Dr. Dai, Dr. Ketani, that you just hear for a second before in the presentation. I work with professor in psychiatry and OB because we are very interested. The main topic right now, the lab, is depression during pregnancy and the gut microbiota. So we work also with experts in nutrition. We also work with people in nursing related with preterm birth, with gestational diabetes, and also with experts in the gut microbiota like Jack Gilbert. So the main thing of the lab is really we are very committed to bring equity and improve women's health using systems biology approaches. So why, as an engineer, we care about that, apart from the importance, right, that women's health it is because from the point of view of an engineering, it's a very dynamic process. It's very, very complex, and it changes over time a lot. Since you are a little girl in this case, until you go into the menopause, it's a very changing time, and it's due to the changes in hormones. And if you just look at the pregnancy per se, in order, for instance, for a pregnancy to work good, to deliver a baby, a healthy baby at the end, there is a lot of things that has to happen. Your immune system needs to change so the baby doesn't die. your endocrine system, the hormones will be released, your brain change. We are now know that the bacteria in your gut also change. And those is a dynamic change over the period of 9 months. So as I say, like all these system has to change at the same time. So you cannot understand it one at a time. You have to understand everything in a holistic way. And in order to do that, you can do, like, at the end of the day, all those changes you can be measuring with different omics approaches. So you can use transcriptomics, proteomics. You can look at intercellular matrix modification that the prior professor also have been talking about. And when any of these processes change or do not work properly, you can have a lot of problems. Like in females, you can have infertility and any other disorders. In pregnancy, that is also the main area of the lab right now, you can have depression during pregnancy, preterm birth, gestational diabetes, and also you can have problems during your menopause. So how we approach this complexity in the lab is by combining by combining multiple different ways, angles of looking at the system, not with us ourselves, but in collaboration with other people that they are experts in the field. So for instance, we use omic techniques, we use observational data, which we recruit for that reason our patients. We collaborate with Dr. Ketani and Dr. Leidan to use in vitro and in vivo models. And then we do a lot of deep learning. And the main thing of the lab will be like trying to how to translate that into something that can be implemented in clinic to help women. So as I mentioned, we have been recruiting women in the clinic. We follow them monthly and we get a bunch of samples from them, like blood, stool, we get urine, whatever kind of fluid that you can imagine. And then we also they get very well phenotyped from diet, from mental health, from exercise, and also we get access to all the electronic and medical records. So now once that you have that, you can look at this thing that it has been coined the gut-brain axis, that is the relationship between the brain and the microbiome, the bacteria that are in your gut. And they talk all the time through little metabolites that bacteria can produce, like serotonin that all of you must have here, that affect your brain. That's the metabolite that works in your brain, right? But also your brain can affect your gut. Think about these butterflies when you have, right, that you have when you get stressed out or worry about something. So in the lab, we have been looking at how these bacteria change over time during pregnancy. And we do see that there are tons of bacteria that change over time in your gut, and you can use it. And those are different in women that they are depressed versus those that they are not. We also look at a bunch of metabolites that we see that they are distinct, exactly as in women that they are depressed, that they are not. One of them has been bile acid, and that has been as motivated us to collaborate with Dr. Kitani, that he's an expert on liver on a chip. But also what the lab does is we also develop new models. So we can get the data that they are out there trying to understand it even better. For instance, in non-target metabolomics is a novel, an approach that they can give you a lot of information of the metabolites that they are there, but 95% of them we have no clue what they are. So we develop also approaches to identify them. The other part that we are doing right now is trying to understand the brain to close this system. And this is Julia, one of our clinical coordinators, trying to show the rest, how to use these portable electroencephalograms and see how the brain changes in women that they are depressed versus not. We are also using the electronic and medical records from patients that we have, and we have seen it, that the difference in women have a substance abuse. If you are carrying a male, for instance, you are more likely to develop repression, which has not been seen before. And now when we have all that information, we can combine it together and develop this dynamic system. So we can combine all the omics, we can combine the environment, and trying to predict which women will develop depression later on. And the other part, and I'm going to finalize with this, is we are also interested in how the pregnancy started. So a lot of women will not be able to conceive regularly for multiple reasons, for disease, but also for treatment during cancer. So we have been able to develop in mice a way to grow follicles, like these small molecules that will transform finally in a oocyte that can be fertilized to make an egg. We have developed a method to do that in mice, and we are very interested in doing it in humans. So right now we have been able to access single cell data. We are developing metabolic models to understand how that happened in real life, in vivo, and how we can use it for women that they have problems with infertility. And with that, I am done.
Okay, I think you should be able to see the full screen now, right?
Yes, thank you.
Okay, please go ahead.
So my name is Michael Strosio, and I'm in the Nanoengineering Research Laboratory in the Department of Biomedical Engineering. I also hold an appointment in Electrical and Computer Engineering, and we have studied the use of nanostructures for biomedical applications. I'm especially interested in a project for an MS student. So next chart, please. So two problems of interest. One is the influence of nanostructures on biological processes. And we've recently looked at spontaneous polarizations from a wide class of quantum dots. Wurtzite quantum dots can produce very large electric fields. We've demonstrated you can switch ion channels, open or closed, by placing these quantum dots near the ion channels. And what we want to do next is look at the effect of placing these quantum dots in electrolytic environments. So this is a theoretical project, computational project. Next chart, please. The other problem is looking at the effect of laser heating of nanostructures and their effect on structures within the cell, such as the microtubule structure or the endoplasmic reticulum, and the release of calcium. And this illustrates our initial studies on a silicon nanowire. Next chart, please. This shows typical heating for a silicon nanoparticle. Next chart. I've listed here current support and where my students have gone over the last half a dozen years. So, and you see the very wide variety of placements after leaving UIC. Again, this is an opportunity for an MS student. Thank you very much.
Thank you, Dr. Storcho. And as you can see, Dr. Storcho has had an illustrious career and placed a lot of students in a lot of different industries. So please do join the breakout room to ask him any questions. Okay, so our next speaker is Dr. Patton. And Dr. Patton, I have your slides. Let me put it up if you're okay with that. So let me do that.
Perfectly fine.
Okay, perfect. So here are your slides.
Wonderful, thank you. I'm trying to be as brief as possible and win the contest on brevity here, because I feel like anybody who wants to can catch up with me either at the breakout or at the by email as this year unfolds here. So my name is Jim Patton. I actually have two existences. I work very hard at the university, of course, but then I'm also part of our nearby hospital research center called the Shirley Orion Ability Lab, which focuses on the idea of what goes wrong with mainly the nervous system after an injury of some sort and how we can maybe fix it. And I come at it from the standpoint of human motor control. So I'm a bit different than I think everybody else that's spoken so far, where I'm focused on control theory and other applications related to human-machine interaction and how we interface with people and machines. So much more gross motor function tasks. What goes wrong with the nervous system and how that affects our abilities to move and act. And then more importantly, maybe sometimes ways to fix it and make it better. So we're using a lot of interesting technology, and mainly our focus is using robotics, but also a bit of feedback technology like virtual reality and other techniques as well. Okay, so let's maybe go to the next slide, and I'll just say a few things. One of the things that I focus on in my, can you advance the slides there, Solomon? Yeah, thank you. So what I thought I'd do here, just for fun, is let's see, there should be a background scene as well. Yeah, that comes up. I'm sorry, it's an animation. But what I've been focusing on in the environment that I'm in is making the best out of being in the middle of an open lab, open clinic, open office scenario, very large set of rooms, many of them actually up and down the different floors of the hospital, all devoted to making sure that everybody involved, including the researchers and the students and the engineers of all types, the opportunity to be in the middle of everything, watching it every day. And that's a pretty exciting opportunity. So we have access to patients, of course, but we also run into patients on our way to get a cup of tea at the watering hole on the other side of the clinic. This hasn't been very great for COVID, but we're coming back from it now. But what's amazing is that all of the questions that you get to ask are from the very first day validated in whether it actually makes sense to ask in the first place, because you're seeing it and asking people of all types whether it actually has any chance of being valuable question to ask in the first place. Us engineers tend to often ask the wrong questions and solve the wrong problems, and spend years doing so before we discover that we should have done something else. And what I also focus on is advanced training and research, and part of that is being able to formulate good questions rather than not just good answers or good solutions to problems. So here's a few brief samples of what we might call proposal briefs. What I ask everybody to do is, if you have an idea, is to try to articulate it in a very small amount of text. So here's some examples of things we're interested in working on in the near future to give everyone an example. And I think I'm going to stop there and just say that, I have about 12 graduate students, and I think right now 3 of those, maybe 4 very soon, will be PhD students, and all of them are also quite approachable and would be willing to chat with you about the experience we have and how we have a lot of fun in our situation.
Yeah, and I think that's a very important point Dr. Patton made, where we have amazing students, as you saw in the small student panel, but each lab has anywhere between 3 to 15 odd students. So please do reach out to the professor. They can put you in touch with their current students, just so that you can get a little bit of a detailed overview of what the whole environment is like. So our next speaker is Dr. Ismail Beghi.
All right, perfect. Hi, everyone. Thanks for joining us tonight. And I I know this has been quite some time, so I'm going to be really fast. I'm going to try my best. I'm Hanana Smalbegi. I'm the director of the Wearable Technology and Sensory Enhancement Laboratory. And mainly what we do is we focus on the movement of the tongue. We want to understand how the tongue moves and how can we use this muscle to our advantage. The main reason we're focusing on the tongue is because your tongue is directly connected to your brain. So unlike your other muscles, it actually bypasses your spinal cord. So in cases of a spinal cord injury, the tongue still retains some of its functionality. So what we set out and we were passionate to do was to take a step towards the communication bill of rights and be able to provide people with disability an assistive device that can help them communicate with our modern life. We are all meeting here today through our computers or interacting through our phones. We wanted to be able to provide that same opportunity for people with disability. So if you're wondering what is really out there for these people, here are some technologies that are actually available in the market, mouth sticks or sip and puff devices that actually do require some movements, residual movements of the neck or the lip muscles. And, you know, we've been hearing a lot about Elon Musk and his really outside the box approach with brain implants, but this brain implants has been around for quite some time in our field, and I've actually come from the background of doing research in this area. While all these technologies are absolutely fantastic and promising, when I set out to create my lab, what we were passionate about more than anything was to develop a device that was wearable, compact, and didn't attract unwanted attention. And we wanted to be able to have a very standalone device that can communicate with any technology out there, anything that accepts Bluetooth. So my team worked really hard over the years, and we developed a compact, smart mouth retainer. What is interesting about our device more than anything is that you can see when it rains, you have a hard time communicating with your cell phone. Now we had to put this device using the same capacitive technology inside the humid environment of the mouth. We had to deal with a lot of saliva. So that's why both on the hardware and software front, we had to do a lot of iterations of development of our device. And now you see Veronica Bratland here, an amazing master's student in my lab, and she obviously used her hands to put the device inside her mouth, but afterwards, she's just swiping her tongue on the roof of her palate, figuring out what are the fun things to do in Chicago. And we've done an extensive human study, and we've realized that after 4 hours of training, subjects are able to achieve 97% of the characters that they type using their finger that we're all used to with their tongue. And our next step is to actually evaluate this device on disabled individuals. And we are extremely lucky because we have great collaboration with Dr. Patton, and he has connected us with an amazing group of speech therapists, and we're using our device as a first of its kind device to study the science behind the tongue motion and understand how the tongue moves and what speed it has, what acceleration it has during its motion, and how can we use that to our advantage in order to be able to quantify speech pathologies. And we're using these to develop models of healthy tongue motions at this point. There are various different applications that we're exploring in our lab, such as using our device for controlling assistive devices, using it for augmented reality, adding various different sensors in collaboration with other faculty here for vital sign monitoring. And if you actually do decide to join our lab, we're looking for motivated master students. We have a lot of fun in our lab. As you can see, once it snowed and we went out and made a snowman and gave it our tongue trackpad device. But in our lab, you get to work both with the hardware and the software part of things, as well as working with human subjects to collect data and analyze that data and make conclusions based on that data. And Here I just have a slide showing the recent graduates from our lab and their placements, and it's been very exciting. The majority of them are working in the area of wearable technology. If you have any questions, I'll be in the breakout rooms. Thank you.
Thank you, Dr. Ismail-Beghi. And Dr. Ismail-Beghi also teaches wearable courses. So if you came to UIC as a grad student, you could also take those courses to see if that's an interesting area for you. So last but not least, we have Dr. Klatt, who's going to talk about his research.
Do you see my slide?
Yes.
Okay. Yeah, good evening. Hello, everybody. So happy to introduce to you the motion encoding MRI lab. So since my research relates to motion encoding MRI, in specific MRI elastography, and this is not a really widely known technique, it's important, I think, to first introduce the basis of this, right? So the basis of this is that manual palpation is one of the most sensitive traditional diagnostic techniques that is applied by medical doctors since centuries, right? So even here in the pyramids, they found these papyrus rolls that are 4,000 years old that basically give instructions on how to use the information obtained by manual palpation in a clinical exam. And because this technique, manual palpation, this traditional technique is so sensitive. And even nowadays, when it comes to cancer, right, I read 30% of all tumors are first detected by touch before any sophisticated imaging technique comes into play. Therefore, it's reasonable and makes sense to develop techniques that visualize exactly what a medical doctor is feeling with their hands when performing a medical a manual palpation exam. So an MR elastography is such a technique that is capable of doing this. So let me just introduce you quickly to the technique, right? There are, in such an MR elastography exam, there are 3 basic steps. First, we have to introduce mechanical vibrations into the tissue of interest. Doesn't really matter how this is done, as long as mechanical vibrations are introduced into the tissue. And then the second step is to acquire snapshots of the propagating mechanical waves using an MRI machine. And then the third step is then to calculate from these snapshots of mechanical waves images of tissue mechanical properties. So the second and the third step that I have here on the slide are visualized now on the next slide. On the left-hand side, you know, on the top for a healthy volunteer, on the bottom for a fibrosis patient, that are snapshots of mechanical shear waves. So the colors encode the in and out displacements in response to an internally or externally introduced mechanical vibration. And you can already see that the wavelengths are different. The lengths of the waves are much longer in the fibrosis patient's liver compared to the healthy liver. And when this is then calculated into images of the shear stiffness, and we clearly see this stiffening of the fibrotic liver as compared to the healthy liver. So what do we do in the motion encoding MRI lab related to MR elastography? So I have in my lab 2 focus areas. One is advanced acquisition approaches, the development of them. And the other focus area is applications of elastography, right? So in order to give the candidates here and potential graduates an idea of what we do, I just present some of the projects. So that was SLIM MRE, or that's what's visualized here. We see there the projections on the 3 coordinate axis of the displacement. Usually these 3 displacement projections in an MR elastography exam have to be acquired in separate steps. And we at UIC developed a technique that is capable of doing this together in one shot. And that's significant. Usually such a scan takes 6 minutes. With this simultaneous acquisition technique, we could reduce this to 2 minutes. So then we build up on that. And this is a current project that my student Jun is working on. DTI, diffusion tensor imaging, is another motion encoding MRI technique. They are not mechanical vibrations are measured, but rather the diffusive motion of water molecules, right, which can also be diagnostically relevant. Membranes hinder the free diffusion of water, or also pathology can have impact on that. It's a different technique. DTI provides completely complementary information compared to MR elastography, but we figured out a way here too to acquire both information types within one scan. So we are basically able to get the DTI information during the acquisition of an MR elastography exam. That's a project where we collaborate with colleagues in Burma at the Beckman Institute, and this technique is currently implemented, and the study is ongoing. That's then testing if the accuracy of the DTI MRE exam is equal to doing this the conventional way in individual experiments. Okay, so the development of advanced acquisition approaches is one focus. The other focus is then to use these techniques and to apply them. Shreyan Majumdar, who just graduated, was involved in this project where we tested, is MR elastography capable of detecting changes in the mouse brain due to the advancement of Alzheimer's disease pathology. And in this study, which was just recently published in Magnetic Resonance in Medicine, we found that the stiffness of the mouse brain while neurodegeneration occurs is reduced compared to the stiffness in the brains of control mice. Another example of application where Rolf Reiter, postdoc in my lab, worked on, that's a collaboration with colleagues from pathology at UIC, where they provided to us human prostate organs just 2 hours after they were basically cut out of the human because they were cancerous, right? So we had there the chance to perform on these samples in such a sample bin. We placed it, performed then basically an MRE exam on these ex vivo organs, calculated maps of the stiffness, and directly correlate with the images that pathology gave to us. And the results that we reported here in this study was that the addition of MR elastography to the standard diagnostic imaging technique for prostate cancer, which is currently multiparametric MRI, the addition of MR elastography information will increase the diagnostic accuracy. We also have a small mini MRI in my lab. This is the size of a shoebox where we can determine the mechanical properties of small samples on the order of 1 centimeter diameter in such a cylindrical tube. I also want to give you the information where, so my lab exists since 2013. The students who graduated, that's where they were placed, in academia and in industry. And then I also want to acknowledge my collaborators and the funding. Yeah, and you are happy to meet me in the breakout room, ask any questions, should you have one, or email me. Thank you.